biochemistry, as you can see in the first slide itself. In the first class, we have completed the monosaccharides and disaccharides. In today's class, we are going to complete the polysaccharides. Fine. So this series includes two classes in which we will complete the chemistry of carbohydrates. Now, before going in the detail of chemistry of carbohydrates, first, let's see what is Auricle and what are the core feature of Auricle. Fine. So Auricle is the platform where you can learn your medical subjects in a very much detail. We are having live classes in the Auricle where you can have one-to-one interaction with the teachers. Then we have a recorded classes as well. So you can watch these recorded classes at your own comfort. Apart from that, we are offering master MBBS books in the terms of notes, and these are the ultimate comprehensive book which will be very useful for the preparation of your medical exams. Apart from that, we also have a MCQs, and these MCQs are from beginner level to the expert level. So from this MCQs, you can assess your level. So that will be helping for the preparation of various entrance examination as well. Apart from that, we have a counseling session as well in the Auricle where you can book your slot for the personalized counseling session where one-to-one guidance will be given. We have a three subscription plans at the Auricle. One is the basic plan, another is the standard plan, and another is the premium plan. In the premium plan, you can have pre-recorded classes, live classes. There is the live doubt clarification, one-to-one personalized guidance is given. Comprehensive notes are also given over there, and you can get the quick revision examination module, which will be very useful at the time of the examination for the quick revision. And if you are using my code, that is Dr. Amit 20, then you will receive discount on your subscription. So that is about the Auricle. Now let's start the second lecture on the series of chemistry of carbohydrate. In the previous class, we have completed monosaccharide and disaccharide. In today's class, we will try to complete the polysaccharides. Fine. So in the previous class, we have discussed that polysaccharides are of two types. Fine. One is the homopolysaccharide, as you can see in the slide, and another is the heteropolysaccharide. So first we will complete the homopolysaccharide and then we will complete the heteropolysaccharide. Now, before going in the detail of homopolysaccharide, first let's see what are the type of the questions which can be asked from this particular topic in your theory examination. So first we will see the example of the questions, and at the end of the homopolysaccharide, we will also see various multiple choice question. So this will be very helpful for your exam preparation. So the first is a long answer question. So the long answer question can be asked from this topic can be describe the structure and biomedical importance of homopolysaccharide. And another type of the question can be asked from the homopolysaccharides will be the short essay question. Short essay questions or short notes. Long essay question can be asked as a 10 marks and short essay questions can be asked as a 5 marks. So the short essay questions can be the compare the structure of glycogen and starch or describe the biomedical importance of homopolysaccharide. So these are the type of the questions which can be asked from the homopolysaccharides. Now let's see the homopolysaccharides in detail. So first, polysaccharides. Fine. So the poly means many. Literal meaning of poly is many, and saccharides means glycan. Fine. So in the Greek word, poly means many, and the saccharides can also be known as the glycans. So polysaccharides are basically composed of hundreds or thousands of monosaccharide units. And we all know that this monosaccharide units, they are connected with the help of glycosidic linkage. Fine. So when there are hundreds or thousands of monosaccharides units are present, that will be known as a polysaccharide. And this polysaccharide can be of two types. One is the homopolysaccharides and another is the heteropolysaccharides. Fine. Or this polysaccharides can also be called as the glycans or complex carbohydrates. Now, this polysaccharide can be either linear in a structure, for example, cellulose, or they can be branched in a structure like glycogen. So that we will see in a detail when we will discuss cellulose or glycogen. Now, what are the properties of polysaccharides? So the polysaccharides are basically high molecular weight substance and they are sparingly soluble in water. and they are not sweetish in a taste. Fine, they are not sweetish in a taste, and they do not exhibit any property of aldehyde or ketone group. Remember, in the previous class, we have discussed that carbohydrates are basically polyhydroxyaldehyde or ketone derivatives. Fine. But this polysaccharide, they do not exhibit any property of aldehyde or ketone group. So this thing should be very clear in your mind. So that are regarding the properties of polysaccharides. Now, as we have already discussed, these polysaccharides are of two types. One is the homopolysaccharides and another is the heteropolysaccharides. Homo means same and hetero means different. This homopolysaccharide can also be known as a homoglycans, fine, and heteropolysaccharides are also known as the heteroglycans. Homopolysaccharides, over there, there will be the same type of sugar units will be there in the homopolysaccharide in the structure, fine. In the structure of homopolysaccharides, same type of sugar units will be there. So that is the homopolysaccharides. While in the heteropolysaccharides, there will be the different type of sugar units will be there. So these are the two types of polysaccharides, homopolysaccharides and heteropolysaccharide. So first we will discuss homopolysaccharide and then we will discuss heteropolysaccharide. So first we will see the what is the biomedical importance of homopolysaccharide. So biomedical importance of homopolysaccharide. So the first importance is they take part in a structural components. That is the first and foremost important. Apart from that, they provide energy to the cells. So they are source of energy. Fine. They provide energy to the cells and they take part in the structural components. So these are the two most important significance of homopolysaccharides. Apart from that, homopolysaccharides like cellulose, they play a very important role as a dietary fiber. Apart from that, they are also useful as a plasma expanders. Fine, whenever there is a hypovolemic shock like condition, they can be used as a plasma expander. Then they can be used as a baby foods as well. And certain homopolysaccharides like inulin, they useful for a determination of nephron function. So this is the biomedical importance of homopolysaccharides. Fine. And dietary fibers, we have already seen, they play a very important role as a dietary fibers. So these are the biomedical importance of homopolysaccharides. Now, first homopolysaccharide is the starch. So first we will discuss the starch in a detail. Fine. So basically starch is the storage form of carbohydrate which is present in the plants, and it is present in a nature in a very abundant amount. Fine. So that is regarding the introduction of the starch. Now, what is the structure of starch? So starch is a basically homopolysaccharide which contains the glucose. Fine. There is a presence of glucose in the starch. And it has a two component. One is the amylose and another is the amylopectin. Remember, amylose is the linear one and the amylopectin is the branched one. So in the next slide, we will discuss what are the differences between amylose and amylopectin. So in the starch, there is a presence of amylose as well as amylopectin. Now, what are the sources of starch? So as I already mentioned that starch is present in a very abundant amount in the nature. So they are present in the plants, fine. And the rich sources are the cereals, fine, particularly the wheat, rice. Then they are present in the potatoes and legumes. So these are the sources of starch. And this is the slide which explains the differences between the amylose and amylopectin. Now, this table is very important, fine, because it can be asked as a viva question. Fine. In the viva question, examiner can ask that what are the difference between amylose and amylopectin. Fine. So the amylose is basically minor component of the starch. One characteristic is it is a minor component of starch. It is present in around 50 to 20%, while the amylopectin is present in around 80 to 85%. Fine. Another characteristic of amylose is it is a water soluble, while amylopectin is a water insoluble. Fine. Then, as I have already mentioned, amylose is the linear one. Fine. And the glucose units in the amylose are linked through the alpha 1,4 glycosidic bond. Remember this thing. It is very important. Amylose, there is a presence of alpha 1,4 glycosidic bond. While in the amylopectin, there is a presence of 1,4 as well as 1,6 glycosidic bond. This 1,6 glycosidic bond will be present whenever there is a branching is present. Fine. Remember the branches of tree. Fine. So whenever the linear chain is there, over there glycosidic bond will be the alpha 1,4. But whenever there is a branching point is there, at that time the glycosidic bond will be the 1,6. Fine. Apart from that, this amylose, when it reacts with the iodine, iodine test is the test which is done for the detection of starch. So this amylose will give the blue color, while amylopectin will give the reddish violet color. Fine. And this amylose has a low glycemic index, while the amylopectin has a very high glycemic index. So these are the differences between amylose and amylopectin. This is the structure you can see. Fine. This is the amylose. Fine. And the glycosidic linkage where present is alpha 1,4. While in the amylopectin, whenever there is a branch is present, over there, there will be the presence of 1,6 linkage. So in the amylopectin, apart from alpha 1,4, one more linkage is present, that is 1,6. While in the amylose, only 1,4 linkage is present. And this amylose and amylopectin both are present in the starch. The minor amount is in the form of amylose, while in the more amount, it will be present in the form of amylopectin. Now, what are the functions of starch? Fine. So as we have discussed, starch is the storage form of carbohydrate in the plants. Fine. So they provide energy to the cells, as well as they take part in the formation of various structural components. Fine. So it is a major source of energy, providing more than 50% of the energy of the cell. So, and it is a storage polysaccharide in the plants. So that is a function. Now, what are the reactions given by the starch? So as I mentioned, iodine is the test which is done for the detection of the starch. So in the iodine, starch will give the blue color, and that is because of the presence of helical structure of amylose. So that is about the reactions of the starch. And starch doesn't answer the Benedict test. Remember, in the last lecture, we have discussed that Benedict test is the test which is done for the detection of the reducing sugar. Remember, again, I'm mentioning it. Fine. Benedict test is the test which is done for the detection of the reducing sugar. Reducing sugar are the substances which have a free aldehyde or keto group. Fine. So whenever there is a free aldehyde or keto group, that type of the sugar will give the Benedict test positive. But in the starch, there is no presence of any pre-aldehyde or keto group, so it will not give the Benedict test positive. So this thing is important. Fine. Starch will not give a Benedict test positive. Apart from starch, sucrose will also not give the Benedict test positive, because sucrose is a type of disaccharide which is a non-reducing disaccharide because it doesn't have any free aldehyde or ketone group. So again, it can be asked as a Viva question, or it can be asked as a multiple choice question. So that is about the reactions of the starch. Now, another important homopolysaccharide that we will discuss is the glycogen. Fine. Till now, everyone is clear. If there is any confusion, doubt, you can ask in the comment box. Okay, fine. So now glycogen. So brief introduction regarding the glycogen. So in the previous slide, I have mentioned that starch is the storage form of carbohydrate in the plants. But in the animals, storage form of carbohydrate is the glycogen. Fine. In the animals, storage form of carbohydrate is the glycogen. So whenever you are consuming excess carbohydrate, it will be first stored as a glycogen. Fine. Where it will be stored? So it will be stored in the two tissues. One is it is stored in the muscle, and another it is stored in the liver. Fine. So these are the two main tissues where this glycogen is stored. And the structure of glycogen is more branched and compact as compared to the starch. Fine. Structure of glycogen is more branched and compact as compared to the starch. So let's see the glycogen in detail. So the structure, so the glycogen, it is a type of homopolysaccharide. Again, it contains glucose. Fine. It is a highly branched polymer of glucose. As we have already discussed, it is more compact and branched as compared to the starch. So this is the structure of glycogen. You can see this is the more branched and compact as compared to the starch. You can see these are all are the branches. And currently already one live classes are going on a biochemistry on Auricle, that was on a glycogen storage, glycogen metabolism and glycogen storage disease. So you can watch that class on the YouTube as well. So for in that class, you can have a more idea regarding the glycogen metabolism. Fine. So this is the structure of glycogen. So you can see this is the more branched structure as compared to the starch. And this middle molecule is known as a glycogenin. So glycogenin is basically polypeptide which is required for the synthesis of glycogen. Fine. So more detail about the glycogenin we will discuss when we will start the series on carbohydrate metabolism. Fine. So this is the simple structure of glycogen. As you can see, these are the glucose units which are joined together with the help of alpha 1,4 linkage. And in the branching point, there is a presence of 1,6 linkage. Fine. So this is the branching point. This red color one is the branching point. And this is the glycogenin, which is also known as the glycogen primer. It is basically polypeptide, which is required to initiate the glycogen synthesis. Fine. So this is the structure of glycogen. Now, what are the reactions given by the glycogen? So when you will give the iodine test for the glycogen, it will give the red color. Fine, it will produce the red color and it is a water insoluble one. So that are the reactions for the glycogen. Apart from that, again, it will give the Mollis test positive. Remember in the last lecture, we have discussed that Mollis test is the general test for the carbohydrate. Almost all carbohydrate give Mollis test positive. Fine. So it answers the Mollis test because it favors the formation of furfuras. Fine. It will not give Benedict test. Fine. Because again, there is a presence of very few reducing glucose molecules. So starch, sucrose and glycogen will not give Benedict test. Remember, Benedict test is the test which is done for the detection of the reducing sugar. And in the laboratory, we do Benedict test for the quantitative estimation of sugar which is present in the urine. Fine. So these are the about the reactions given by the glycogen. Now, what are the sources of glycogen? So it is a storage polysaccharide which is present in the animals and it's stored in the liver as well as in the muscle. Fine. In the liver up to 5% and the muscle it is present in the 1%. Apart from the liver and muscle, it is also stored in a various tissues like brain, intestine, kidney in a smaller amounts. But the majorly it is stored in the liver as well as in the muscle. Now the glycogen which is stored in the liver, it has a certain role and the glycogen which is stored in the muscle, it has a certain role. So whenever the blood glucose level gets decreased, fine. After taking the food, blood glucose level will be very high. But after 2 to 3 hours, the blood glucose level will start getting lower. Fine. So how this blood glucose level is maintained? So this blood glucose level is maintained with the help of the glycogen which is stored in the liver. So whenever the blood glucose level decreases, this glycogen which is stored in the liver undergoes degradation. Fine. It will get broken down. It will get broken down, glucose will be released and it will restore the blood glucose level. So the glycogen which is present in the liver, it plays very important role in the regulation of blood glucose. Fine. This is the one important point. And the glycogen which is present in the muscle, it plays important role in the muscle contraction. So whenever the energy needed for the muscle contraction, that energy will be given by the muscle glycogen. So these two things you should be knowing. Fine. These two things are very important. Fine. So the breakdown of hepatic glycogen provides mechanism for the blood glucose regulation. Fine. Hepatic glycogen plays very important role in the regulation of the blood glucose, and the muscle glucose provides energy for the vigorously contracting muscle, say for doing exercise. Fine. So this role of glycogen which is stored in the liver is different from the role of glycogen which is stored in the muscle. Muscle glycogen doesn't play any role in the regulation of blood glucose. Remember this thing. And why they are not playing important role in the regulation of blood glucose? Because doesn't have a one enzyme that is known as a glucose 6 phosphatase. Glucose 6 phosphatase. Fine. What is the role of this enzyme? That detail we will discuss when we will start the series on carbohydrate metabolism. But right now, remember that muscle doesn't have a glucose 6 phosphatase enzyme. That's why muscle doesn't play any important role in the regulation of blood glucose. Muscle glycogen only play important role in the muscle contraction. So that is about the functions of glycogen. Now, hydrolysis. So glycogen, when it will be hydrolyzed by the enzymes like salivary amylase and pancreatic amylase, fine. So it will produce the various products like maltose, Fine, there is a production of maltose, then there is a production of maltriose, and then there is a production of limit dextrine, and there is a production of traces of glucose. So these are the end products of hydrolysis of the glycogen. It will produce maltose, maltriose, limit dextrine, and traces of glucose. So that is about the hydrolysis of glycogen. Now, what are the disorders associated with the glycogen? So any enzymes deficiency related to the metabolism of glycogen will result in a one particular disorder, which falls in the category of glycogen storage disease. Fine. This again is very important question that we will be discussing in a metabolism of carbohydrate. Fine. So inherited deficiency of enzymes for a glycogen metabolism leads to the glycogen storage disease. So till now we have discussed definition of homopolysaccharides, what are the properties of homopolysaccharides, and two important homopolysaccharides. One is a starch, which is a storage form of carbohydrate in the plants. Then we have discussed glycogen, which is the storage form of carbohydrate in the animals. Now in the next slide, we will discuss the other homopolysaccharides. So the first homopolysaccharide is a inulin. Again, inulin is very, very important. So many multiple choice questions asked from this particular topic. Fine. Inulin is very, very important for the multiple choice questions. Fine, so let's start. So again, inulin is a type of homopolysaccharide which is made up of polymer of fructose. Remember this thing. It is made up of fructose units. All starch and glycogen, they all were made up of glucose unit. But inulin is a homopolysaccharide which is composed of a fructose unit which are linked by glycosidic bond. Fine. Remember, it is made up of fructose unit. Fine. And the role of inulin is, it is very, very important for the estimation of glomerular filtration rate. It is used to measure the GFR in the form of inulin clearance test. Fine. The sources of inulin are, it is present in the various plants such as onion bulb. It is also present in the garlic. It is also present in the tubers. That is the sources. And uses, I have already mentioned, it is used to measure the GFR, that is glomerular filtration rate, in the form of inulin clearance test, since it is not metabolized by our body. So that is the use of inulin. The question which is frequently asked from the inulin is, it is a fructose, a polymer of fructose. Fine, it is a fructosan. And another, what is the use of inulin? So it is used to measure the GFR. So that is the inulin. This is the now another important homopolysaccharide, which is again very, very important, is the cellulose. So cellulose is basically non-starch polysaccharide. Cellulose is basically non-starch polysaccharide. It is a non-starch polysaccharides. Fine. And cellulose, again in the cellulose, there is a presence of glucose unit. It is a polymer of glucose unit, but this glucose units are joined together with the help of beta 1,4 linkage. Remember, in the cellulose, there is a presence of beta 1,4 linkage is there. Fine. So this beta 1,4 linkage are present in the cellulose. Fine. So that is the structure of cellulose. Now we don't have any enzyme which can digest this beta 1,4 linkage in our body. Fine. So basically we don't have an enzyme which is known as a cellulase or cellobiase. Fine. The name of enzyme is the cellulase. Another name is the cellobiase. So that's why human cannot digest the cellulose. Fine. So whenever we take cellulose in the diet, it is not digested by the human beings, because we don't have a enzyme which can digest this beta 1,4 linkage, and the name of enzyme is the cellulase or cellobiase. So that's why this cellulose plays very, very important role as a dietary fiber. Remember this thing, cellulose play very, very important role as a dietary fiber. Fine. So dietary fiber are the substances which provide roughages to the stool. Fine. Apart from that, they regulate the peristaltic movement. They prevent the constipation. They prevent the colon cancer. Fine. They reduces the glucose level. They reduces the cholesterol level. Dietary fiber has got so many importance. And the cellulose is the main part of the dietary fibers. Apart from the cellulose, the dietary fiber also includes agar, pectins, all these substances. But the main part of the dietary fiber are the cellulose. And the cellulose is not digested by the human beings, but still they are very, very important because they provide roughages, fine roughages to the stool, they regulate the bowel movement, they prevent the colon cancer, they prevent the constipation. So these are the significance of dietary fibers, fine. And why they are not digested? Because we don't have a beta 1,4 glycosidic linkage. So remember this thing regarding the cellulose. Fine, we don't have a cellulase enzyme. While herbivorous animals, they have a cellulase enzyme, so they can digest the cellulose, but humans cannot digest the cellulose. Fine. So it helps in a peristaltis, helps in a softening the stool, relieve the constipation. So these are the significance of dietary fiber. So this cellulose can be asked as a short answer question in the form of dietary fiber. It can be asked in the form of dietary fibers. Fine. So in that, whatever I have mentioned, that you have to mention. So cellulose is a part of dietary fiber and it is a non-starch polysaccharide. Remember, it is a non-starch polysaccharide. Now, another one homopolysaccharide is a dextrine. Fine. Another homopolysaccharide is a dextrine. Fine. So dextrine, again, it is produced by the partial hydrolysis of the starch. Whenever we do the hydrolysis of starch, if it is a partial, at that time, the product which will be synthesized is the dextrine. So in the dextrine, the glucose polymers are present. Fine. And these polymers are connected with the help of alpha 1,4 and alpha 1,6 linkage. Fine. Glucose units are linked by alpha 1,4 and alpha 1,6 glycosidic linkage. And depending upon the molecular size, it will give either red color or violet color with the iodine. So that is dextrine. Fine. Sources is they are obtained during the partial hydrolysis of the starch by the enzyme amylase. Now, what are the uses of dextrine? So dextrines are useful for the preparation of baby foods like Cerelac. Fine, they are useful for the preparation of baby foods like Cerelac. Apart from that, they are also important for the formation of various paste like material that are known as a mucilages. Fine, so that are the uses of dextrine. So they are used for the preparation of baby foods as well as they are useful for the preparation of paste. So that is a dextrine. Now another one is the dextran. Fine. Dextrine is different and dextran is a different. Fine. So in the dextran, again, there is a presence of glucose polymers, but these polymers are connected with each other with the help of alpha 1,4, alpha 1,6 and alpha 1,3 linkage is there. Remember the additional linkage over here is the 1,3 linkage is there. Fine. In the dextrine, only alpha 1,6 and alpha 1,4 linkage is there. But in the dextran, there is alpha 1,4, alpha 1,6 and alpha 1,3 linkages there. Now, how this dextran is prepared? So dextran is prepared by the action of bacterial enzyme on the sucrose molecules, fine, which is present in the sucrose medium. So that's how this dextran is prepared. And this dextran has got very important biomedical importance. They are used as a plasma volume expander. So whenever there is a hypovolemic shock, which can occur in a various condition which in which there is a extreme blood loss like road traffic accident, like in the case of burns. So there will be the hypovolemic shock. So in hypovolemic shock we need a plasma volume expander. So this dextrans are useful as a plasma volume expander. So that is the use of dextran. Fine. Now don't get confused with these matching words, that is dextrose, dextrine and dextran. Dextrose is basically glucose only. Fine. As glucose is a dextrorotatory, so in the clinical practice it is known as a dextrose. Fine. Dextrine we have already discussed. It is a intermediate which is synthesized in the hydrolysis of starch. Dextran we have already discussed, which is used as a plasma volume expander. So that are the some rhyming words, dextrose, dextrine and dextran. So don't get confused. Am I clear till now? If there is any confusion, doubt, you can always put in the comment box. I will be very happy to answer it. Fine. So till now we have discussed starch, glycogen, which are the main homopolysaccharides. Then we have discussed the other homopolysaccharides in the form of inulin, which is a polymer of fructose, which is useful for the estimation of GFR. Then we have discussed Dextrine, fine, which is used as a mucilagase, which is used in the paste formation. Then we have discussed dextran, which is used as a plasma volume expander. Now another homopolysaccharide is a chitin. Fine, chitin. Fine. So chitin, it is a present in the exoskeleton of various insects like crabs, cockroaches. Fine. So it is made up of N-acetyl D-glucosamine units which are linked by alpha-1,4 glycosidic linkage. It is made up by N-acetyl D-glucosamine. So basically it is made up by the amino sugar. Fine, which is linked by the alpha-1,4 glycosidic linkage. And it is present in the exoskeleton of various insects like crabs, lobsters, cockroaches. Fine. So that was all about the homopolysaccharides. Now let's see the some multiple choice question which is asked in a various entrance examination. You can answer the question in the comment box. Fine. So the question is cellulose is. Question is cellulose is, and the options are complex lipoprotein, starch polysaccharide, non-starch polysaccharide, or complex glycoprotein. I will wait for a 1 minute. You can answer the correct answer in the comment box. Cellulose is a complex lipoprotein, starch polysaccharide, non-starch polysaccharide, or complex glycoprotein. So the correct answer I have already mentioned that cellulose is the component which is used as a dietary fiber, which is not digested by the human beings because we don't have a cellulase enzyme, and it is a non-starch polysaccharide. It is a non-starch polysaccharide, and that is the correct answer. So correct answer is C. Then another question is glycogenin is, and again it was asked in a NEET PG 2019 exam, and the options are it is a polypeptide, polysaccharide, lipid, or glycosaminoglycan. Fine, again I will wait for a 1 minute. You can write down the correct answer in the comment box. Glycogenin, we have already discussed it. Fine, glycogenin is, which is also known as the glycogen primer, which is required to initiate the glycogen synthesis. So glycogenin is basically polypeptide. Fine, the correct answer is it is a polypeptide. So correct answer is A. Another question is starch is synthesized by animals, plants, both or none. Please write down the correct answer in the comment box. I will wait for 1 minute. Starch is synthesized by. Fine. Starch is a homopolysaccharide. Fine. Which is very found in a nature in a very abundant amount because it is present in the plants. It is a storage form of carbohydrate in the plant. So the correct answer over here is the B. So the correct answer is B. Then another question is the following polysaccharide is the chief constituent of dietary fiber. And the options are starch, glycogen, cellulose, or none of the above. So the answer of this question again we have discussed in a detail in a last 2 slides ago only. Fine. So what will be the correct answer over here? The following polysaccharide is the chief constituent of dietary fiber. So the chief constituent of dietary fiber is the cellulose. Fine. Other dietary fibers are the agar, pectins. They are present as a dietary fiber, but the chief constituent of dietary fiber is the cellulose. Remember, in the cellulose, glucose units are joined together by beta-1,4 glycosidic linkage. Fine, that's why we are not able to digest it. So the correct answer is C. Another question is the following is used in measuring GFR, and the options are cellulose, inulin, starch, or none of the above. Fine. Again, I will wait for a 1 minute. Which one is used to measure the GFR? Again, this question is from the homopolysaccharides. Cellulose is a homopolysaccharide, inulin is a homopolysaccharide, starch is a homopolysaccharide. So the correct answer over here is the inulin. Inulin is the homopolysaccharide which is used to measure the GFR, and it is made up of polymer of fructose. It is made up of fructose. Fine. So that is the correct answer. So correct answer is B. Now, so that was all about the homopolysaccharides. Fine, with the example of the theory questions as well as example of multiple choice question. Now we will discuss the heteropolysaccharide. And again, this topic is most IMP for your theory exam, because from chemistry of carbohydrate, if any question is asked, then that question will be the heteropolysaccharide. So that's why you need to read this topic by heart. So let's start the heteropolysaccharides. Fine. So first we will see the type of the questions which can be asked as a theory exam, which can be asked in a theory exam. So it can be asked in a theory exam as a long answer question in the form of discuss composition of mucopolysaccharide and glycosaminoglycans with their function. So this can be asked as a 10-mark question. Fine. Or it can be asked as a short essay questions in the form of describe the structure of glycosaminoglycans or describe the biomedical importance of glycosaminoglycans. And as I already mentioned, it is a most IMP question from chemistry of carbohydrate. So let's start heteropolysaccharide. So heteropolysaccharide, as we have discussed, they contain different type of glucose units. And over here, different type of glucose units are mainly present in the form of disaccharides. Fine. It is made up of repeating disaccharide units of different monosaccharides and their derivatives. And the example of heteropolysaccharides will be the glycosaminoglycans, agar and pectins. So these are the three examples of heteropolysaccharide. Remember, the three examples of heteropolysaccharides are glycosaminoglycans, agar, and pectins. These are the examples of heteropolysaccharide. While the example of homopolysaccharides are starch, glycogen, inulin, chitin, dextrin, dextra. Fine. While the example of heteropolysaccharides are glycosaminoglycans, agar, and pectins. Fine. So let's discuss all these three examples in a detail one by one. So first we will see the glycosaminoglycans. Now this glycosaminoglycan, again, it can be asked as a short note like glycosaminoglycan itself. The question will be like glycosaminoglycan itself. So this glycosaminoglycan, they are also known as a mucopolysaccharide. Another name of glycosaminoglycan is a mucopolysaccharide because they were found in a mucous membranes. So it is known as a mucopolysaccharide. Fine, as they were detected in the mucous membranes. So this glycosaminoglycans, as we have discussed, they are made up of repeating disaccharide unit or sugar units. Fine. And this repeating disaccharide unit will be present mostly in the form of amino sugar and uronic acid. So in the mucopolysaccharide, there is a presence of amino sugar plus uronic acid. And this two will be linked by glycosidic linkage. Fine. So remember, mucopolysaccharides structure will be, there will be the presence of amino sugar. So amino group is attached to the sugar. So it will be the glucosamine or galactosamine, or there will be the presence of uronic acid. Uronic acid is the acid which is produced by the oxidation of sugar. Fine. When sugar will be oxidized, it will result in a formation of uronic acid. So let's see this glycosaminoglycans now. Fine. So amino sugars can be the glucosamine, acetyl glucosamine, or an acetyl galactosamine. Fine. So these are the examples of amino sugars. And the example of uronic acid or sugar acid will be the D-glucuronic acid or iduronate acid. These are the example of sugar acids. Apart from that, there will be presence of sulfate group. This sulfate group, either it will be attached to the amino sugar or it will be attached to the sugar acid. Remember, the structure of mucopolysaccharide are amino sugar and uronic acid. In the amino sugar, it will be present in the form of glucosamine or N-acetyl glucosamine or N-acetyl galactosamine. Fine. When amino group is attached to the glucose or sugar, it will result in a formation of amino sugar. And apart from amino sugar, there is a presence of uronic acid, which are the sugar acid. And this uronic acid will be either glucuronic acid or iduronate acid. Fine. And the sulfate group will be attached either to the amino sugars or sugar acids. Now, what are the example of this glycosaminoglycans? So again, I will ask you to remember the name of this glycosaminoglycans, because all glycosaminoglycans are important, and all this glycosaminoglycan we will discuss one by one. So the first glycosaminoglycan is hyaluronic acid. First one is the hyaluronic acid. Second one is the heparin. Second one is a heparin. Third one is a heparin sulfate. Fine. Third one is a heparin sulfate. Then fourth one is a dermatan sulfate. Fifth one is a chondroitin sulfate. And the last one is a keratin sulfate. So these are the example of glycosaminoglycans, which are also known as a mucopolysaccharide. So till now, everything is clear. If it is clear, then I can go ahead. If you have any doubt, then you can uh, please put down in the comment box. So I will be very happy to answer it. Okay, fine. So let's go ahead. Fine. So these are the example of glycosaminoglycans. That is hyaluronic acid, heparin, heparin sulfate, dermatan sulfate, chondroitin sulfate, and keratin sulfate. Now let's discuss all this one by one. So first, hyaluronic acid. So first we will see the structure of hyaluronic acid. So we have seen the general structure of mucopolysaccharide, which is that presence of amino sugar and uronic acid. So in the hyaluronic acid, there is a presence of D-glucuronic acid in the form of uronic acid, and there is a presence of N-acetyl glucosamine in the presence of amino sugar. Fine, and both this will be joined together with the help of beta-1,3 glycosidic linkage. Fine, they will be joined together with the help of beta-1,3 glycosidic linkage. Fine, so that is the structure of hyaluronic acid. And one more important feature of hyaluronic acid is it doesn't contain any sulfate group. It is the only glycosaminoglycan which doesn't contain sulfate and not attached to the any protein. So it can be asked as a multiple choice question in your various entrance examination. Fine. Which glycosaminoglycan doesn't contain sulfate? And the option will be hyaluronic acid, heparin, heparin sulfate, and chondroitin sulfate. So the correct answer will be the hyaluronic acid. It doesn't contain any sulfate group. So this is the structure of hyaluronic acid. Fine, there is a presence of glucuronic acid. This is the glucuronic acid, and there is a presence of N-acetyl glucosamine. You can see there is a presence of hydroxyl group over here. Fine, CH2OH. So that is the amino sugar. Now, what are the sources of hyaluronic acid? So hyaluronic acid is basically present in the synovial fluid. Fine, remember the main place where this hyaluronic acid is found is the synovial fluid. Apart from synovial fluid, it is also present in the vitreous humor, and it is also present in the outer coat of mammalian eggs, and it is also present in the extracellular matrix of loose connective tissue. Fine, it is present in the mainly in the synovial fluid, which is present around the joint cavities. Apart from that, it is present in the vitreous humor and the outer coat of mammalian eggs. And what is the function of this synovial fluid? So synovial fluid act as a lubricant. It is present in the synovial fluid. Over there, it acts as a lubricant. So it prevents the friction between two bones. Fine. So it prevents the friction. Fine. Apart from that, it also acts as a shock absorber. Fine, it acts as a lubricant, it acts as a shock absorbance, and it is very effective barrier against the bacterial invasion. Now the hyaluronic acid, which is present in the outer coat of mammalian eggs, fine, it will be digested by one particular enzyme which is present in the seminal fluid, and that enzyme is the hyaluronidase enzyme. The name of enzyme is the hyaluronidase enzyme. Fine, so the hyaluronic acid which is present in the outer coat of the mammalian eggs, it is digested by the hyaluronidase enzyme. Fine, it is digested by the hyaluronidase enzyme. Fine. So by that, it will be playing very important role in the fertilization. Fine. So it will enable the penetration of the sperm and the fertilization of ovum. Apart from that, it plays very important role in the cell migration. Fine, during the morphogenesis. So that is the significance of hyaluronic acid. The main significance is it acts as a shock absorber as well as it acts as a lubricant, and it is present in the synovial fluid as well as in the vitreous humor. And the thing that you have to remember for the hyaluronic acid is it doesn't contain sulfate group. Fine. So that is a hyaluronic acid. Now again, another important mucopolysaccharide is the heparin. Till now, it is clear everyone. If you have any doubt, then please put down in the comment box. Okay, let's start with the heparin now. So heparin, again, a very important mucopolysaccharide. Fine. First, we will see the structure of heparin. So in the heparin, again, there is a presence of uronic acid in the form of idiuronic acid sulfate. Fine. And there is a presence of amino sugar in the form of D-glucosamine sulfate, which are linked by beta 1,4 glycosidic linkage. Fine. Over here, there is a presence of idiuronic acid sulfate. Remember this thing. Now, sources of heparin. So, heparin is present in the various tissues like lung, liver, spleen, and it is present in the mast cells and it is present in the circulation. And it is the mucopolysaccharide which is a intracellular. Remember, it is a intracellular one. While all other are the extracellular, this heparin is the intracellular one. So, it is a naturally occurring anticoagulant which is present in the lung, liver, spleen, skin, and mast cells. Now, what is the function? So we have already seen it is a naturally occurring anticoagulant. So it will prevent the clotting by interacting with the antithrombin 3. Fine. Apart from that, it also helps in a clearance of clearing the plasma by activating the lipoprotein lipase enzyme. So whenever we receive the turbid plasma, that turbid plasma will be cleared by the lipoprotein lipase. Fine. It helps in clearing the turbidity of lipemic serum. Lipemic serum is the serum which will be there when you will give the blood immediately after taking the food. Fine. So this heparin will help in the clearing the turbidity of plasma by activating the lipoprotein lipase enzyme. Now, what is the clinical significance? So, clinical significance is it is used as an anticoagulant in various clinical condition for which are prone for creating the thrombus, for prone for presenting with the thrombosis, like myocardial infarction. It is given in a myocardial infarction to prevent the thrombosis. So that is the clinical significance. So this is the picture you can see. This is the heparin injections which are used for in a myocardial infarction to prevent the thrombosis, and these are the heparin vacuoles. Fine, these are the green color vacuoles are the heparin vacuoles. They are the heparin vacuoles, which contains sodium heparin. Fine, so they are useful for the clearing the turbidity of plasma, as well as this heparin vacuoles. They are useful for the ABG estimation and serum electrolytes. In the clinical biochemistry laboratory, there are different type of vacuoles are there. Fine. Plain vacuoles, irritative vacuoles, fluoride vacuoles. So one of them, it is a heparin vacuole, which is a green color vacuole, and it is useful for the estimation of ABG as well as for the serum electrolyte. So that was about the heparin. Now another one is the heparin sulfate. So first, again, we will see the structure. So the structure is uronic acid is D-glucuronic acid or idiuronic acid sulfate, and the amino sugar is the N-acetyl glucosamine sulfate. Fine, so that is the structure of heparin sulfate. And heparin sulfate contain higher acetylated glucosamine residue than the heparin. Now, it is present in the basement membrane, particularly in the glomerular basement membrane of nephron. Fine. It is present in the basement membrane of the nephron, as well as it is also present in the cell surfaces. So what are the function of this heparin sulfate? So it is function as a structural component. Apart from that, as it is present in the glomerular basement membrane of nephron, so it prevents the selective loss of proteins. Fine. It prevents the loss of proteins. So it acts as a barrier. Fine. It acts as a barrier to prevent the passage of plasma protein. Fine. To prevent the passage of plasma protein. Fine. So that is the function of heparin sulfate. Apart from that, it also involved in a cell-to-cell communication, cell growth, and cell differentiation during the morphogenesis. Now, what are the disorders associated with the heparin sulfate? So there are two disorders which are known as a mucopolysaccharidosis. One is the Hunter's syndrome and another is the Hurler's syndrome. In detail about these two syndrome will be discussed in a carbohydrate metabolism. So two disorders are associated with the heparin sulfate. One is the Hurler's syndrome and another is the Hunter's syndrome. Now, dermatan sulfate. So dermatan sulfate, as the name suggests, it will be present in the skin. Fine, and the structure of dermatan sulfate is it contains idiuronic acid and N-acetyl glucosamine. Fine, so that is the structure of Dermatan sulfate. Sources is, as the name suggests, it is present in the skin. Apart from that, it is also present in the blood vessels and heart valves. So that are the sources of dermatan sulfate. What are the functions of dermatan sulfate? So the function of dermatan sulfate is it is playing very important role as a structural component, fine, in the matrix of various connective tissue, as well as it maintain the shape of the tissues. Now, what are the disorders associated with the dermatan sulfate? So again, two disorders which are associated with the dermatan sulfate is the Hunter syndrome and Hurler syndrome, same as that of heparin sulfate. Hurler syndrome is caused by the deficiency of lysosomal iduronidase enzyme, and Hunter syndrome is caused by the deficiency of lysosomal iduronic acid sulfatase enzyme. Both are the mucopolysaccharidosis. So that is about the dermatan sulfate. So till now we have discussed hyaluronic acid, which is present in the synovial fluid, fine, which doesn't contain any sulfate group, which plays very important role as a shock absorber as well as the lubricant. Fine, then we have discussed heparin, which is acting as a natural anticoagulant and it is the only intracellular mucopolysaccharide. Then we have discussed heparin sulfate, which is present in the glomerular basement membrane. Then we have discussed dermatan sulfate, which is present in the skin, blood vessels, and the heart valves. Now, keratin sulfate. So the keratin sulfate, again, it is a very important mucopolysaccharide which doesn't contain any uronic acid in the structure. In the keratin sulfate, there is a presence of only amino sugar, and this amino sugar will be present in the form of N-acetyl galactosamine sulfate, and there is a presence of D-galactose. And this too will be connected with the help of beta-1,4 glycosidic linkage. Remember, in the keratin sulfate, there is no uronic acid is present. There is no uronic acid. Most IMP multiple choice question. Fine. Which mucopolysaccharide doesn't contain uronic acid? So the correct answer will be the keratin sulfate. And this keratin sulfate is present in the cornea, fine, as well as it is present in the tendons, bone, and cartilage. Fine. But mainly it is present in the cornea. Now, what are the functions? So the functions of keratin sulfate is it plays important role as a structural component. It will be aggregated with the chondroitin sulfate. Fine, and it is also responsible for the transparency of cornea. Fine, as it is present in the cornea. Disorders which is associated with the keratin sulfate is the type 4 mucopolysaccharidosis, that is known as a Morquio syndrome. So that is a disorder associated with the keratin sulfate. Now, another one is the chondroitin sulfate. Chondroitin sulfate, the structure is over here, there is a presence of D-glucuronic acid in the form of uronic acid, and there is a presence of N-acetyl galactosamine sulfate in the presence of amino sugar. And the sources is it is a widely distributed mucopolysaccharide, fine, which is present in the various cartilages, synovial fluid, tissue, tendon, bone, heart valves. Fine, it is present in the connective tissue, cartilage, bone, and heart valves. Fine, so the function will be, it will be playing important role as a structural component. Fine, and it may be aggregated with the keratin sulfate. Then another, so that was all about the glycosaminoglycans or mucopolysaccharide. Now another heteropolysaccharide is the agar. Fine. So agar is the polysaccharide which is made up of glucose or galactose unit. Fine. It is a polymer of sulfated galactose or glucose molecule. And the source of agar, it is originated from the seaweed. Fine. It is originated from the seaweed. And the uses of agar. So agar, basically, it is useful for the preparation of various culture media because it is not digested by the bacteria. In the microbiology, you can see there is a preparation of various culture medium to grow the bacteria. Fine. So this agar is used as a support medium. Fine, over there. Apart from that, this agar is also used as a support medium for gel electrophoresis because this agar has a particular characteristic. So whenever you will heat the agar, it will be the liquefied. And whenever you will cool down the mixture, it will produces the gel-like structure. So that's why this agar is used in a gel electrophoresis. So that are the uses of agar. Apart from that, I have already mentioned that this agar is also important dietary fiber. Apart from cellulose, this agar is also important dietary fiber because it is not digested by the human beings. So it is used to relieve the constipation. So it is used in a microbiology lab for the culture medium and as well as it is also useful for the gel electrophoresis like immunodiffusion and immunoelectrophoretic technique. So that is about the agar. And the now last mm, uh, heteropolysaccharide is the pectin. Again, pectin is also used as a dietary fiber. So let's see what is the structure of pectin. So pectin is made up of galactose and uronic acid residue. Remember over here there is a galactose. There is no amino sugar is there. There is only galactose and there is a presence of uronic acid residue. And it is present in the various fruits, apples and carrots. What are the uses of pectins? So pectins are used as a dietary fiber, as I already mentioned. Cellulose, agar, and pectins, they are used as a dietary fiber. Apart from that, they are also very useful for reducing the blood sugar level as well as the blood cholesterol level. They are also useful for the preparation of various jellies-like material in a food industry. So that is about the pectin. So this is all about the polysaccharides. Fine, particularly the heteropolysaccharide. Now let's see the multiple choice question. So first question is glycosaminoglycans are, and the options are A, extracellular matrix, ground substance in the connective tissues, mucopolysaccharides, or all of the above. So let's wait for a 1 minute. Please write down the correct answer in the comment box. Glycosaminoglycans are, they are extracellular matrix, ground substance in a connective tissue, mucopolysaccharides, or all of the above. What will be the correct answer? So the correct answer is it is present in the extracellular matrix. So the option A is correct. They are present as a ground substance in connective tissue. That is also correct. They are also mucopolysaccharide. That is also correct. So the correct answer is D, all of the above. So the correct answer is D. Second question is, all of the statements are, sorry, actually, I'm not able to read it. All of the statements are correct with respect to the heparin, except, and the options are homopolysaccharides prevent conversion of prothrombin to thrombin, anticoagulation or prevents the deep vein thrombosis. So you have to find out the incorrect statement for the heparin. So please mention the correct answer in the comment box so I can have idea. What will be the correct answer? Okay, all of the statements are correct with respect to the heparin. So the correct answer over here will be the homopolysaccharide. Heparin are heteropolysaccharide. They are not homopolysaccharide. So the correct answer is A. Second question is glycosaminoglycan, which is present in the cornea. And the options are keratin sulfate, hyaluronic acid, chondroitin sulfate, and dametin sulfate. So please mention the correct answer in the comment box if you know the correct answer. Glycosaminoglycan, which is present in the cornea. So the correct answer will be the keratin sulfate. It is present in the cornea, which is responsible for the transparency of cornea, and keratin sulfate doesn't contain uronic acid. Remember this thing, it doesn't contain uronic acid. So that is about the another question. Then mucopolysaccharide that doesn't contain any uronic acid residue. And the options are heparin sulfate, heparin, chondroitin sulfate, or keratin sulfate. So I have already discussed this in the previous slide itself. So the correct answer is D, that is keratin sulfate. Fine, mucopolysaccharide which doesn't contain uronic acid is a keratin sulfate. Another question is activity of enzyme lipoprotein lipase is increased by, and the options are hyaluronic acid, chondroitin sulfate, heparin, or all of the above. Lipoprotein lipase is the enzyme which clears the turbidity of plasma. So that will be activated by which substance? Hyaluronic acid, chondroitin sulfate, or heparin. So the correct answer is heparin. Heparin will activate the lipoprotein lipase. So the correct answer is heparin. Then another question is synovial fluid of the joint contains heparin, hyaluronic acid, dametin sulfate, or all of the above. So what will be the correct answer? So synovial fluid of the joint contains hyaluronic acid. It acts as a shock absorber as well as the lubricant. Apart from synovial fluid, it also present in the vitreous humor of the eye. So the correct answer is B. So that is all about today's class. If you have any confusion and doubt, then please mention in the comment box so I can have idea regarding it. And you can follow the Auricle on the Instagram page. as well as you can subscribe the Auricle channel on the YouTube for very interesting videos. We are posting very interesting videos related to the clinical pulse, related to the mnemonics, fine. And you can also access all these live classes on the YouTube channel as well. Fine. So that is all about today's class. In the next class, we will be starting another important topics related to the biochemistry. Updates about it, you can get it on the official Instagram handle, that is Auricle_official. So that is all about today's class. Thank you. Thank you.